questions. Whoops, I hadn't meant to do that yet, but now it's done. So I encourage people to unmute themselves during the seminar to ask questions, or if you feel that it would be better to ask questions via chat, that is great too. We'll try to make sure that all the questions get asked. So it is my great pleasure to introduce today's CSMR seminar speaker, Connie Chang from the Department of Chemical and Biological Engineering at Montana State University, where she's also a member of Montana State's Center for Biofilm Engineering, which is actually sort of worldwide, I guess, the place for biofilm engineering. It's really very impressive indeed. So Connie got her PhD from UCLA, where I guess you worked with Tom Mason, right? Because I know that you had this sort of like a lot of hits with nano emulsions. So I followed that with a whole lot of interest. And then on from UCLA to Harvard, where she did her postdoc with Dave Waits. And so that involved innovations in making microemulsions with microfluidics. I should say emulsion droplets with microfluidics, so that you could then have any kind of chemical environment or biological environment you want inside of the droplet. And so so this is work that again, we at the CSMR had become aware of as it was going on and heard about it and learned a little bit about it when Connie came to visit us just sort of informally about a year or so ago, when we realized we really had to get her here to New York to tell us all about it. The pandemic intervened, but at least we sort of said, okay, yeah, but we can get her to talk about the microenvironments inside droplets anyway. And of course, the pandemic, though, is short-circuiting that, as you'll see. So you'll have to come back again after the pandemic when you can come back in person. So the thing about studying biofilms in these microenvironments is that you really can, for the first time, isolate the biofilms and perform experiments on them to understand how they work as a colony, how they work as a community, how they evolve, not just how the individuals evolve, but how the biofilm itself evolves. It's a very, very powerful platform. And I guess this work is pretty heavily funded. I know that Connie received a career award for this from the NSF. It's very exciting work. And I guess we're not going to hear a single word about it because instead, what we're going to hear about is how a physical scientist can dive into the COVID pandemic and make important contributions that'll help to get us out of this crisis. So with that, I will turn it over to Connie.
Thank you so much, David. It was such a pleasure to visit you all last year, and I had a great time. And so sad that this pandemic happened, but here we are. So today I'm excited to talk to you about Lab on a Chip and diagnostic tools for COVID. Recently, ever since the pandemic hit, my lab has transitioned over to studying COVID. So I'll tell you a little bit about where we are. And so I guess I don't have to really introduce coronavirus, the cause of this global pandemic. I guess one of my projects as a postdoc was being able to understand viral evolution to prevent pandemics. And it's ironic because I guess I never really expected an actual pandemic to happen, but here we are. And so as you know, the COVID-19 comes from the SARS-CoV-2 coronavirus. The reason why it is called coronavirus is because if you take a side profile of the actual virus, it looks as though there's a crown. And that is the capsid protein that surrounds the nucleic-acid protein and the RNA that's encapsulated inside of this inside of this virus. So it's a single-stranded RNA virus. It has one of the longest RNA genomes of an RNA virus, about 30,000 bases. It's, corona is the Latin word for crown. And so you can see here from a side profile, it kind of looks like a crown. And actually, if you look here under TEM, we have grown this virus here at Montana State University. and imaged under the TEM, transmission electron microscope, and you can see little spike proteins coming off of these individual virions. I guess it's well known for causing the common cold, and there's alpha and beta strands of coronavirus, but it's also known for causing more dangerous viruses, such as that came out in 2003, and also MERS, which came out in the Middle East in 2012 from camels. And so recently, this virus, SARS-CoV-2, was first. I guess it first came out. I think it bats. Can you still hear me? Okay.
Yes, you're fine. I have no idea what's going on. I am not doing that.
Okay, sounds good. I was wondering if you can all hear that. But anyway, so in December of 2019 is when this SARS-CoV-2 came out, and it causes the disease known as COVID-19. Yes, so the virus, and we've been growing up this virus here at Montana State University. We have a biosafety level 3 containment facility called the BSL-3 laboratory, and it's located here off campus. I'm maybe, see if I can get my pointer. I'm located right here, and the BSL-3 laboratory is here, located close to the veterinary diagnostic laboratory. You actually need to be highly cleared to work in such a laboratory. You need FBI clearance, and you have to go through a lot of training to be able to get in. When the pandemic hit, both my laboratory, a virologist laboratory on campus, a herpes virologist, and an immunologist all started working on this virus. As you can see, you need a lot of personal protective gear to work in there. So full-on suits and PAPRs. But this allows us to work with the virus. And so we started out in March and began to grow up the SARS-CoV-2 virus. We grew them up in vero cells. And so vero cells they're African monkey fibroblast cells. And they're really well known for amplifying all sorts of viruses, such as polio and rabies, influenza. So they're pretty susceptible and will allow for viral amplification. So the vero cells is what we started off with. And you can see here that there's a difference just from imaging when you look at uninfected cells here on the right versus infected cells. So the infected cells kind of have the cytopathic effect where the virus lyses the cell open, and these cells start to appear white and dense, which is indicative of infection. So one of the really great advantages of having a BSL-3 laboratory is the ability to perform viral infectivity assays. And so I think that this is really like a bonus. My virus collaborator, my influenza collaborator at UIUC does not have a BSL-3. So, he's not able to work with this virus. So in the BSL-3, what you can do is you can perform 2 general types of these infectivity assays. One is the plaque assay. And that is where you have maybe a 6-well plate, and you plate these wells with cells. And then you take a dilution series of your virus, and then you put the virus on top of the cells. Now there's an agarose overlay on top of that, so that when the virus infects a cell, it can infect neighboring cells. The virus can diffuse to the neighboring cells, but can't go beyond that. So what ends up happening is you have these bright spots here after the plaque is negatively stained, and these individual plaques are indicative of one infectious virus that has infected a single cell. So you count the number of plaques, and virologists use this all the time. It's really common, and it kind of tells you the concentration of viruses in terms of plaque forming units, which is these spots that you see, these clear spots as of over milliliters. Another form of the plaque assay is called the Endpoint titration assay. It's also often called TCID50, and it's where 50% of these cells can become infected with the virus. And usually these 2 numbers correspond really well with each other. I think virologists tend to perform and prefer the old school plaque assays, but either can be used to detect for viral infectivity. So this is just a beautiful image taken from our BSL-3 laboratory. And here is actually the host cell, the vero cells I told you about. They're stained, the nuclei are stained blue with a DAPI stain. And the green is actually antibody with tagged with a green fluorescent protein. And this antibody can attach to the nucleic-acid protein of the SARS-CoV-2 virus. So it's very specific to the nucleic-acid protein of the virus. And you can see here, these are cells that have been infected with the virus, and you have viral proteins here that are detected through this antibody and replicating inside of the cell. And you can see just in this like neighboring cell right here, that's where infection is starting to happen, but it's not, the cell is not fully infected yet. So just a beautiful image in terms of being able to see and visualize viral infection. Okay, so you guys, I don't know how many of you have gotten the actual CDC assay or been tested for COVID-19, but in order to, I haven't gotten it yet, and I've heard it's not very pleasant. So typically what happens is you have this 6-inch long swab that's inserted into the cavity. Like I've heard it's a brain tickle here. And you have this nasopharyngeal swab that's inserted into literally the back of your head. I'm not sure if you'll be able to hear this video. I can hear it, but you may not, but I'll just walk you through it. So the patient blows her nose. Her head is tilted backwards. Her eyes, you ask the patient to close her eyes so that the swab can be put into the nasopharynx. It's rotated a few times for 10 to 15 seconds. And then the swab is placed into the container. And I've heard that you have to also have it done in both of the nostrils. So, so this is the typical test that would be done. You have that long swab and that's inserted. Okay. And I don't know if this happened in New York. Here we had a huge swab shortage at the beginning of the pandemic, and then recently, just maybe about a month ago, we're still running out of these swabs. And I feel like the supply chain is a difficult issue. You know, we're trying to figure out if we can access different types of. I think we have to try to get whatever tubes that we can in order to do these saliva tests. So the supply chain is a huge issue. And in the beginning of the pandemic, we started printing a lot of these swabs. You can use an autoclavable resin. We have several of these Form Lab printers in my colleague, Dr. James Wilking's lab here at MSU. And you know, that's what we ended up doing. So when we ran out of swabs was 3D printing a lot of them. And then once you have the swab, you also need trained personnel to administer this test. And so, you know, you can get a test through drive-through, but you do need someone that's in full-on PPE gear testing you. You do the nasal swab, and then you put it into this tube that contains a viral transport media. So the viral transport media is a media that you can really make very easily in the laboratory, and here's the formulation right here. You know, it contains a little bit of nutrient. It contains antibiotics, antifungals, and it really allows you to place that swab in and preserves the virus in there for 72 hours until you're ready to test the sample. Here's an example of a virologist, the Langlois lab at University of Minnesota, who made a lot of these 15 ml tubes with 3 ml of the viral transport media in them. So you have your swab, you have your viral transport media, and now if you perform the gold standard RT, which stands for reverse transcription, qPCR, quantitative PCR test. So this is the gold standard, and this is the most sensitive assay, the commonly performed assay to figure out if you have a COVID-19. So you have your sample that's collected, put into viral transport media. The first step that you have to go through is RNA extraction. So like I showed you in the first slide, you have your virus, which has your capsid proteins and your envelope around the virus. But you have to access the single-stranded RNA that's on the inside of the virus. And so you perform RNA extraction, which typically in the lab takes about an hour. And it involves a lot of these steps, these short centrifugation steps, where you have a binding buffer that binds the RNA. You spin it down and you add washing buffer so that you really just isolate this purified viral RNA in a small amount of solution. So this tends to be a little bit pricey, adds time, but is the standard way of accessing the viral RNA. Then you take that viral RNA and you mix it with your qPCR mix. So this is your real-time PCR mix that has all of the primers, has a reverse transcriptase in there, it has a DNA polymerase in there, and it amplifies for the viral RNA. You fill up your well plate and you put it into quantitative PCR machine. This is all done in about a 2-hour process where you use reverse transcription to turn your RNA into double-stranded cDNA. And then from there, you have your DNA polymerase that starts to form more of these double-stranded DNA that then get detected through a fluorescence signal. So if you're positive, you have your cycle. So it goes through several cycles, which I'll talk to you about. But if you're positive, your fluorescence intensity will cross over a threshold here. And that's how you know you're positive for the virus. And so these cycle numbers go from 0 to 40, where you're cycling between temperatures 40 times. If you're positive, again, it crosses over that threshold. And if you're negative, it never crosses that threshold. So it's a very, a little bit of a yes or no answer, but you can also tell, which I'll tell you about, the reason why it's called quantitative is because depending on where it crosses this threshold, it tells you about the concentration of the original virus you have. Okay, so a little bit more about the reverse transcription quantitative PCR. So you have, again, your target viral RNA. You have your primers. The reverse transcription step takes half an hour at 60 degrees to turn it into double-stranded cDNA. And now you undergo the cycling. So you denature it close to boiling for 15 seconds, which turns it into single-stranded DNA. And then it undergoes primer annealing and extension, which is 60 degrees for 1 minute. And here what we have is we have a TACMan probe. And that's a fluorescent probe that tells you every time this amplifies, you'll have a fluorescent probe every time your DNA is extended. And that tells you, that increase in this probe tells you how much is amplifying over time. So you measure this FAM fluorescence, which comes from the TAC probe, and you do this over 40 cycles. And so what you can see here is you can see the FAM probe, which is your fluorescent probe. You have a ROX probe, which is a way to normalize your signal. So you take this normalized fluorescence signal, and again, like I said, when it crosses over this threshold value, that tells you the concentration of RNA you have. So if you have high amounts of RNA, you will cross over the cycle threshold much sooner than if you have low RNA. So here you can see the cycle number is 10, where it crosses and then increases as you go to lower amounts of RNA. And you could make a standard curve here, which is the cycle threshold as a function of your log base 10 of the RNA concentration. And from that slope, you can figure out an efficiency of your reaction. A good efficiency of reaction is at 100%, but it can range between 90 to 110%. Okay, so this is RT-qPCR. We often just in the laboratory call it qPCR. Okay, so this is the gold standard method, the Cdc assay. It consists of actually 4 different primer sets in the CDC emergency use authorized qPCR test. So you're amplifying for 4 different genes here. The N1, N2, and N3 all come from the nucleic-acid of the virus. And finally, you have a human ribonuclease P gene. So this human ribonuclease P gene is important when you're doing clinical testing because it tells you whether or not you're human. So that has to be positive. And if it's not positive, then your test is usually inconclusive because you don't know where you could be, say, sampling RNA from the subway in New York City, something like that, off of a surface. So that tells you whether or not you're human. And then the N1, N2, and N3 tell you whether or not you have the viral gene there. And recently, because the N3 didn't work very well, or since the beginning of the pandemic, the N3 didn't work very well. So right now it's these 3 gene segments in the official CDC test, the N1, N2, and the human ribonuclease P gene. Okay, so this is a laborious process, and just to give you an idea of how long this takes, we did transfer one qPCR machine to our local hospital to be able to increase up to 100 samples a day. And so 100 samples a day takes 3 batches of extraction of the RNA extraction method I told you about, and then it takes 3 technicians to run this particular qPCR test by hand. So quite a laborious and lengthy process. In my lab, I think in the beginning of the pandemic, there were some problems that we saw with the CDC assay, and you saw that they took away the N3 one. And so we went with the WHO primers, the World Health Organization primers, and we amplified for the RDRP and the E gene segments of the virus. And here you can see that we were able to successfully amplify and detect up to 10 to the 8th viruses using our primers that we developed, and also we had very good efficiency in our qPCR reactions. So this is what we're using now moving forward in my laboratory. Okay, so probably you have all heard about the importance of rapid testing. There was a period of time here in Montana in the summer when it was taking up to 2 weeks to get your result back. And by the time the 2 weeks were over, you had already quarantined yourself for 2 weeks. So there was really no reason to have gotten the test. And so a lot of the research is now saying that rapid tests, having more rapid tests is more important than having a very sensitive test such as qPCR. And so you can actually see here the limit of qPCR is down to 1,000 copies per milliliter. So that's here 10 to the 3 in terms of a viral load per milliliter. I often think about that in terms of microliters. And so I think of that as 1 copy per microliter is pretty much the limit of quantitative PCR, qPCR. And so if you're looking here, yes, you can detect virus from somebody that is maybe exposed after 2 days of exposure. And so it is a highly sensitive test here, but a lot of argument is being made in terms of having a rapid test that can detect the virus at maybe a higher viral load, may not have the sensitivity as qPCR, but it's still good enough to detect if you are transmissible for the virus, which you are between, say, 3 days to 9 days after exposure. And so this is at like about 10 to the 5, about 100 copies per 10 to the 5 copies per milliliter, about 100 copies per microliter. Okay, so it's more important with the rapid test to be able to test kind of the upswing of a person who is infected, so that you can rapidly then take action in terms of quarantining and contact tracing. And this paper here from Roy Parker and Michael Vina also talks about how it's not the test sensitivity is not as important in terms of frequency of testing in terms of the pandemic. And so if you look here, if you're looking at the difference between, say, a 10 to the limit of detection of 10 to the 3, which would be your qPCR test, versus a limit of detection of 10 to the 5, maybe a different rapid test such as LAMP, the differences are negligible in terms of, you know, the percent infectiousness removed. So if you test daily, you can remove most everybody. Every 3 days is also pretty good at, you know, close to 80 to 90% of removing infectious people. Weekly starts to drop, and of course 14 days, it's just, you don't have a test that's effective.
Excuse me.
Yes.
Viral load is the number of viral particles per microliter.
In this particular case, it is per milliliter.
Per
Yes. Mm-hmm.
It's the number of viral particles per milliliter.
That's right.
Okay, thank you. Sorry to interrupt you. Thank you.
No, thank you for interrupting. Happy to answer questions. It's difficult because I can't see anybody, right? So, yeah. So I do have my chat box open if you want to put in any questions there as well. Okay, so back to rapid testing. That's why I became very excited about LAMP. Because it's so easy and it's so fast. I'll tell you a little bit more about it. So I told you about this polymerase chain reaction, which was invented in 1985. It's slow, it's expensive. You know, the CDC test itself is probably $25 a test. You're tied to a laboratory, and you need an expensive piece of equipment, a qPCR machine, quantitative PCR machine, to do the temperature cycling. So that machine probably costs anywhere between $50,000 to $90,000. And so, you know, it's great, and it's a workhorse, and everybody uses it. But I guess nobody has ever had to test anybody, to test like the quantity of people and just so quickly to get those results back, right? So that's why I think that LAMP is a really it's a newer type of amplification of nucleic acid. It was invented in 2000. It's very rapid. I'll tell you about how fast it is. Within half an hour, you'll have a result. It's very cheap, probably about $1 an assay. It uses really basic equipment. And so all you need, because it's isothermal, it's held at one temperature, 65 degrees. You just need a basic heater. So maybe a water bath, an oven. I've seen papers using a sous vide machine, things like that. Single temperature, very field deployable. So you can take this outside in the lab. You're not tied to an expensive machine in the lab, and it's a robust reaction. So again, like I mentioned, it's fast. It probably takes 30 minutes versus 2 to 4 hours with qPCR, 4 hours if you have to extract and do a lot of samples, 2 hours if you're just doing qPCR. It's simple, again, isothermal, one temperature at 65 degrees. It's very cheap, $1 an assay. It has been used for rapid field testing of really dangerous viruses such as Zika, Ebola, and dengue. And I think the reason why there's so much interest in it now is because of its speed and its cost. I think where it can really shine is maybe not in the diagnostic world, but in more of a surveillance screening purpose, where maybe you can use this to be able to rapidly test a huge population of people, and then say from that huge population of people, like pick out the needle in the haystack, the one person that might be infectious, that might be asymptomatic and shedding virus, but may not know that they're sick, that they have the virus. And so I think that this could be a really useful screen, because it is so fast, to be used at points of entry, nursing homes, workplaces, schools, allowing you to screen thousands a week. And so here's an example of Dave O'Connor. He's a virologist at the University of Wisconsin-Madison. And here you can see that he's just has all these, he has a back of the van type of test. He has LAMP in the back of his van, and he's testing the entrance of a school right then and there. Chris Mason, he's actually in New York at the Weill Cornell Medical School. Is that how you say it? Weill? Cornell Medical School in New York City. Anyway, so he is a geneticist there, and he grew up in Waukegan, Wisconsin, and so he helped bring LAMP testing to the firefighters there in the city. And so here he is in the firefighting firefighter station. You know, has a little lamp set up here. Okay. Yes. So Dave, you asked a question. If qPCR takes 2 hours per measurement, how do labs analyze 100 samples per day, multiple machines? Yes, you need robotics. So that gets expensive, right? So we did a lot of quotes on different types of robotics where, you know, you can automate everything, and they can cost up to $100,000 per robot. And I think that's the only way that you can do high throughput. It's like this high throughput liquid automation. They have cheaper robots, which we've also looked into, called Opentrons. So if you're interested in like a really cheap liquid handler, they're just probably $5,000 and you can program them. And yeah, I think that we might go that route if we do need a liquid handler. Okay, so why LAMP? Again, no, you don't need an experienced personnel to run LAMP. It is very, very simple. You don't need expensive equipment. And the great thing about LAMP is it's colorimetric. So it's very easy to see your results outside of the laboratory. You just heat up your tube and you can see color changes by eye. So here you can see the LAMP reaction in my laboratory where we have pink and it's the negative control. If you're negative, your samples are pink. If you're positive, your samples are yellow. And so here we spiked in the RDRP and E genes of the SARS-CoV-2 and also the HA gene of influenza. And so they're all positive here. And I like this paper. I call it the coffee cup paper, even though it's titled SARS-CoV-2 on-the-spot virus detection directly from patients. My colleague here, Asaf Rotem, who I worked very closely with as a postdoc in the Waits Lab at Harvard, is also on this and worked on this with his friend from high school, I think. And so it's as simple as this. So you spit into a tube. Now this tube has a little bit of a chemical called proteinase K. This chemical breaks up the RNases and DNases that are present in saliva that can prevent amplification of the RNA. And so you incubate your saliva sample with this proteinase K for about 15 minutes at room temperature. And then what you can do is you can heat inactivate the virus at a very high temperature for 5 minutes. This heat inactivation step makes it so that you can handle the saliva safely. And it also releases the RNA from the capsid so that you can amplify for the RNA. So then you heat and activate it, then you take a small sample through a loop, and this takes exactly 1 microliter of sample, and then you put it into your LAMP mix, and that's that pink mixture which contains primers, an enzyme, nucleotides, and then you put it into your coffee cup. So this coffee cup is set. You can see here, I chatted with Asaf on Zoom, and he showed me his kitchen experiment, which I'd love to see. But you have here a temperature strip, and you can set the temperature so it's at 65 degrees in this coffee cup, and then you just stick your tube in there. And if it's positive, it'll turn yellow. If it's negative, it's negative. If it's pink, it's negative. It says here this method is currently under development and research and is not approved as a self-usage for COVID-19 validation to test. But here is Asaf, and he's testing himself and his family weekly with this test in this coffee cup. So I love it. I guess you can test at home, but if you are so lucky as to have a yacht and to own a yacht, you can also perform LAMP testing on a yacht. So this is Jonathan Rothberg, and it says, this was a wonderful article actually in the New Yorker. It was like fascinating and fun to read. So it says here, while quarantining abroad his yacht, Rothberg has devoted himself to developing a diagnostic test that is as fast, simple, and inexpensive as a home pregnancy test. Yeah, so he's kind of a serial entrepreneur. Actually, he started, I think he was one of the co-founders with Dave Waits on starting Braindance. At least that's what I heard. And so, here he is on his yacht performing LAMP, and he wants this to be as easy as, brushing your teeth in the morning or putting on a pot of coffee. You can also just take a LAMP test. And so this is his assistant. He has all of these devices here. And you can see here is, it's really as simple as I think he's testing it with just a simple heat block. So yes, testing aboard your yacht. What a luxury. So why saliva? I think I started becoming really interested in saliva over the summer, and I kept telling my vice president of research here that we should start doing saliva. I was getting all of these like emails from my collaborators at both Rutgers and UIUC how they were doing these saliva tests and had these kits. I think Rutgers ended up commercializing it into Volt Health. And UIUC now, we just talked to them last week, they're up to testing 10,000 samples a day. It costs $1 million a week to do this, but they have 17 stations on their campus, and they have these golf carts where they transport these 50 milliliter tubes all the way to the veterinary center where all these tubes are being processed through liquid handling. So it's amazing. It's just amazing to see their operation. They're down to 0.1% positivity. And as I kept talking about saliva, the Yale Saliva Direct test became FDA EUA approved. The UIUC test also became EUA approved. And so I think finally my administration was like, okay, let's do your saliva test. So Yale, was sponsored by the MBA, which is pretty fun. And it's, as I'll tell you, go through this, but it's a very simple method, their Saliva Direct method. So saliva, so easy, no extra swab and no uncomfortable brain freeze type testing. Okay, so why saliva? This article came out in the New England Journal of Medicine. And it turns out when they did the study of looking at nasopharyngeal swabs compared to saliva, it correlated really well. And actually it was found that more viral RNA was detected in saliva compared to swab. In addition, here, there was a higher percentage of saliva samples compared to nasopharyngeal swab samples that were positive for up to 10 days after the COVID-19 diagnosis. And so so I think that this was a really kind of a turning point, maybe in August, where people started to think about saliva, and I'm hearing that a lot of universities are turning to this. So we started this saliva work in my laboratory. I've never done a human studies subject before, so that was interesting going through our institutional review board. And so I said, okay, let's try a pooled sample, and everyone spit into a 1 ml Eppendorf tube using a 1 ml tip. We took our pooled saliva, we added that proteinase K chemical, we combined it and we mixed it. And then we heat and activated for 5 minutes at 95 degrees. Added a small sample of that to our lamp mixture, and then heated it for 30 degrees at 65 degrees. And so this is what the test looked like. It's simple, a color change. It's based upon the lamp test. And here are some of our results. So we tried, we spiked in the SARS-CoV-2, both the extracted RNA, and that's the extraction process I walked you through with the spin steps. And then we also tried heat inactivated virus that came out of our BSL-3 laboratory. So to work with the virus, you can work with the live virus in the BSL-3, but if you have a BSL-2 laboratory like I do, it needs to be heat inactivated and tested for infectivity before you can pull it out of the BSL-3 for testing. So we did this heat inactivated virus as a surrogate spike in control. And you can see here, these are the results that we got both in water and in saliva. So on the left here, we have a no template control. So when we just put in water, nothing amplified, and the solution remained pink as expected. If we put in a positive control, which is an N gene segment, all of these tubes turned yellow. So that's also as expected. Now the R-actin is that test is a gene that's similar to the RNase P in the CDC assay that tells you whether or not you're human. So actually, if you look in all of these saliva samples, it's all yellow. And that makes sense because we're testing a human sample, saliva. In water, however, sometimes half the times the solutions, the tubes stayed pink and half the times they turned yellow. And so what this is telling me is that, yes, we did not have the cleanest tubes. And, you know, we're not a clinical testing lab in my laboratory, so there's probably human R-actin on the surfaces of these tubes. Okay, but this is the real test, which is the last column here. And so if you take a look at the SARS-CoV-2 RNA that we spiked it in at 10 to the 7, 10 to the 5, and 10 to the 2 copies per milliliter. It turns yellow in both the RNA and in saliva. So we can really detect down to the 10 to the 2 copies per mil. And here you can see in saliva, though, we're having saliva is not as sensitive. And so there's something about the saliva background that doesn't allow you to go to such low limits of detection. And so we weren't not able to see it at this lowest level of detection. But still good enough to test for infectivity. And you know, that corresponds really well with our 10 to the 5 viral load here. And we'll be able to test if you're virus transmissible. So we're excited. We just got this grant on the 17th of October, $776,000 from our wonderful Governor Bullock here. And so we will be using this to explore LAMP for surveillance testing. We have a van here that we'll be using. It's called Herb. And we're now gearing up to be able to, compare LAMP with the CDC assay I told you about, and also a saliva direct assay, which we will get up and running as well. So we really are interested in comparing qPCR, saliva directly to qPCR, and saliva directly to LAMP. Okay, so I told you a lot about the colorimetric assay for LAMP, and but there's a lot of research in terms of developing LAMP for other technologies. So this is the method I told you about, where you can actually lyophilize your LAMP reagents here and then add your sample, add a liquid sample, and then look at the color change. You could put the LAMP reaction on a lateral flow device, and so you can also test this way. It tends to not be as sensitive as, say, the tube-based method, or my area of research here at MSU, which is laboratory on a chip. So I'll tell you a little bit more about my area of research in microfluidics. So in my lab, we work a lot. We've been working a lot in the past few years on influenza virus and studying influenza using microfluidics. And so in microfluidics, you're manipulating really small amounts of fluid from the picoliters to the microliters in small volumes that you can manipulate on a chip that fits in the palm of your hand. So it really allows for a really quick sample to result turnaround, because you've miniaturized everything onto a small volume, into small volumes. It tends to be more sensitive and specific, affordable, using much smaller amounts of reagent, and rapid. And so you can see here at UIUC, they've developed a microfluidic chip that's based upon LAMP. Paper microfluidics, that's the basis of the lateral flow. And here, this is a chip that uses pumps and valves and actuators to manipulate the liquids on a chip. And that has been actually commercialized by Fluidigm. And now Fluidigm is offering a microfluidic saliva test. So my area is in drop-based microfluidics, and where you can think of drops as tiny picoliter scale microreactors. And here you can see single cells being encapsulated into drops. So they're separated by oil. You typically need a biocompatible surfactant here to stabilize the drops to prevent them from coalescing. And again, it's biocompatible, so you can put all sorts of biological materials into the drops, whether it's single cells. We routinely put in there single host cells for the virus, DNA, RNA, proteins, viruses, enzymatic substrates. The size scale of these drops tend to be about the width of a single piece of human hair, so somewhere between 10 to 100 microns in diameter. Now, you need a high-speed camera to view these drops being formed. And so we've taken a short video and slowed it down for you to see here the fluid dynamics on the chip. But you can create thousands of drops per second. So it's really at this kilohertz rate. Here you can see single cells being encapsulated into drops. Here they are all collected into a tiny test tube in the laboratory. And in this test tube, you probably have about 1 million drops. You can take this test tube, you can incubate it in a cell culture incubator, and then you can say re-inject them into another chip if you want to analyze the fluorescence from the drops or do any downstream drop manipulation, such as adding small amounts of liquid to each drop that passes by these electrodes. So these electrodes destabilize the drop, allow you to perform what we call pico injection. You can do the opposite, where you can extract a small amount of drop, say from a long slug. This is taking off 1% of the drop from this long slug, or say 10% of a drop. And finally, we have this very powerful technology that allows you to sort drops based upon their fluorescence. And so you can see here the drops that are dark have a fluorescent dye in them. The drops that are light do not. And you can detect this very rapidly and then be able to sort for fluorescent drops at kilohertz rates. So that's what we do routinely in my laboratory, and we work a lot, like I mentioned, with influenza virus. And so I told you a lot about qPCR. Well, we've been able to do qPCR within drops. And so here you can see our bulk dilution series in qPCR. This red curve here is 10 to the 9 copies per microliter, very high concentration. In drops, it corresponds to 10 to the 4 copies per drop, so 10,000 copies per drop. And, we've gone all the way down to 10 to the 4 copies per microliter, which corresponds to 0.1 copies per drop. So this is what it looks like in drops. And so after you've amplified, these are endpoint fluorescence images. These drops here have 10,000 copies in them. These drops here at 10 to the 4 have 10,000 copies in them. These drops here are the ones that are at 1 in every 10, 0.1 copies per drop. So 1 in every 10 drops had a single copy of virus. And the ROX reference dye is in all of the drops, and we use that for normalization. So we have good efficiency in these drops. We can detect down to a single copy per drop. And you can visualize that. We can use fluorescence imaging in order to quantify the fluorescence and to count the drops that are bright. We can also use our high throughput fluorescent detection stand, where we can run these drops along in a microfluidic chip and then detect the number of bright versus dark drops. And so we have a nice Poisson fit with these dilution series, and it's exciting that we can detect down to a single copy per drop. I'm not talking about this in this talk, but the whole reason why we're doing this is one of the main questions that we have is we really want to answer how many viruses does a single infected cell produce? So that's something that I think virologists cannot do at a really high throughput level. Maybe you can do 100 single cell experiments in well plates. You can scale it, but really we wanted to look at hundreds of thousands of cells and look at the distribution of burst sizes. So how many, is it one cell that's producing a lot of viruses, or do all of the cells produce equal amounts of virus? Influenza virologists think you're somewhere between, the burst size is somewhere between 100 to 1,000. So it's a huge range in terms of this burst size, in terms of knowing how many viruses a single cell produce. So that's one of the questions we're really, really interested in answering. Okay, but related to COVID, we're also working on developing a microfluidic device, which I call the Freedom Chip. I think there was a period of time, you guys were probably like this too, when you were quarantined at home. And I just wanted to be free. So I call this the Freedom Chip. And it's a cheap, disposable, rapid point of care microfluidic device that uses lamp and drops. And so we're prototyping this. We're prototyping a chip that can hold the drops at a single temperature. And then we can image the drops being, lighting up under a microscope. And Yeah, so the Freedom Chip is going to allow us to safely gather, work, see friends and family, travel, and allow us to meet in person. So I'm not talking to you across Zoom. So that's the idea here. And, you know, with the first SARS epidemic, or SARS, with SARS-1, it didn't spread, right? I mean, it spread through Asia, but, you know, it didn't, I think there was a lot of worry that it was going to come here, but it wasn't a pandemic. But, you know, what you could easily detect someone that was infected with SARS because that person had a high temperature and a high fever. And so it was pretty easy. You could use, you know, a heat gun, you could use an IR thermal heat camera to be able to isolate people that were infected with SARS. So it was fast, simple, easy, effective. It doesn't work, unfortunately, for this virus, for SARS-CoV-2. And so the idea here is that we want to develop this Freedom Chip that uses saliva, isothermal lamp. As far as we can tell, there are no supply issues with making the lamp reaction. Each person would have their own microfluidic chip where it would be self-contained, disposable, very cheap, very cheap. We think probably under $3 a chip for the entire chip. And it will allow for a really high speed detection of the virus. And so this is the general idea. You would have your chip and maybe a saliva tube together in a bag. You would spit into the saliva tube. You would get that saliva onto the chip, where the chip would be preloaded with your lamp reaction and also your oil. Rapidly make drops and then heat up your chip. So far, preliminary results are showing that it takes a shorter amount of time in drops than it does in bulk to amplify. And then you would take a picture of your chip and get a result by cell phone. And so this is just a kind of a comparison with other rapid point of care tests. You know, there's not very many really accurate ones on the market for nucleic acid. You guys, I think there's been a lot more of these rapid antigen tests, such as the Abbott that you might have heard of, and these very fast lateral flow assays. They tend to not be as sensitive compared to a nucleic acid test such as LAMP or PCR. So I'm pretty excited because I think that, you know, this could be very cheap, very rapid. So maybe perhaps our goal is to shoot for under 5 minutes. So here are some of our preliminary results. We developed primers for the RD, RP, and E genes of the virus. And here we've amplified for concentrations of the RD, RP, and E genes. We can go down to, we've done a whole range from 4 copies per microliter all the way to 40 million copies per microliter. And you can see at the highest concentrations here, takeoff happens rapidly within 5 minutes. It does depend on the concentration of virus, but you know, this whole reaction is done within, I would say 15 to 20 minutes, or maybe 20 minutes. We think that we only need to maybe look at when amplification is starting to occur in the drops. And so, you know, I think that could be as short as 5 minutes. We're working on making this test faster. We can also detect the full length genome within using LAMP. We've done some preliminary work with influenza. One of the goals here is to multiplex to be able to look for both influenza and SARS-CoV-2. Yeah, and this is supposed to be for fall season, but fall is already here. And as a proof of concept, we've shown that it does work in drops, and preliminary results are showing that drops are more sensitive and the reaction happens faster than in bulk. And again, this is you know, one pot reaction that happens just on the chip. So that's kind of where we are now. These are my funding agencies. My lab has grown in the past few years. And if we get the chance to travel again, and if you guys are here and ever come up to Bozeman, Montana, I would love to see you. We are surrounded, it's just a beautiful place. We're surrounded by mountains. And, you know, we wear puffies in the winter, and then we float the Gallatin River in the summers. So I'm happy to welcome all of you if you get a chance to come out west. Thank you.
All right, thank you very much. That's super cool. So this is, yeah, audible claps and everything. So this is open for questions, and I'll kick it off. One of the things that you said a couple of times in your presentation was that the reactions proceed faster in the droplets than in bulk. Why would a reaction proceed faster in a droplet than in bulk?
So yes, that's a very good question. The reason why is because, you know, normally if you, let's say you have one template in a bulk reaction, well, that, you know, amplifies, and it can amplify. It's because of the effective volume. So you have, in bulk, you have a large effective volume, but in drops, you've squeezed your reaction down to a tiny volume. So in a sense, like because your volume has decreased, but your concentration of amplicons inside of this small volume increases at the same rate, that concentrate, it's like your effective concentration is a lot higher in the drops.
So it's that the products don't have time to diffuse away, so you can.
That's right. It's kind of isolated.
I see.
In a small volume. Yeah, so we're seeing, like, maybe in bulk, if something takes about 12 minutes, we've shaved off a few minutes, and we're starting to see amplification around 7 to 8 minutes. Here's some of our preliminary results.
So, what's wrong with this? I mean, it looks like it's the answer. So what?
It does look. It does seem to be the answer, but you know, we've been working on the chip design. Actually, I pulled Dave into this, so his group and my group, there's a group of us all working together and designing the chip itself, right? Like, we don't have, that's, I think, pretty tricky. You know, how do you make it as simple and as cheap as possible and as rapid as possible? You know, do you use, like, say, finger valves to get the liquids onto the chip? Do you use a vacuum manifold? I think there's a lot of ideas that we're going through in terms of the easiest way of making a simple point of care device out of drops. So that, in addition to the assay, saliva is turning out to be a little bit tricky to work with. Like I mentioned, there's a lot of inhibitors in there, and it's not as sensitive as, I think, the swab, the viral transport media of the virus in the viral transport media. So I think there's a lot to optimize, but you know, I do think it's certainly a way forward as a test.
So, are there more questions?
I don't know. I can't seem to stop share.
Oh, I can stop you if you want to share. If you want to stop sharing.
Sure.
Let's see. So yeah. So I will. So whoops. Hold on. I think what I'll do is I'll share my own.
Okay.
And then
great.
I'll stop mine.
Now I see y'all.
Hi there.
Hi.
So okay. So
thank you.
if there are no more questions, I'll stop the recording now.